and Vadodara, and also visiting professor and PhD guide, Swinburne University, Australia, Sheridan University of Science and Technology, Gujarat, and visiting professor at Slovak Academy of Science, Slovakia, Sumandip Vidyapith, Gujarat, and Acharya Nagarjuna University, Andhra Pradesh. Dr. Ramchand was the director at Kemin Pharma, India, and was holding a simultaneous positions as director of research and development, Kemin Industries, South Asia Private Limited, which is a part of Kemin Industries in USA. Dr. Ramchand was also a WW executive team member of Kemin Industries prior to the assignment of Kemin Industries. Dr. Ramchand was with the Sun Pharmaceuticals, currently the largest pharma company in India, as its vice president and head of drug discovery research. He was closely involved in the setting up of a multi-billion drug discovery center, including design and development of laboratories. Currently, Dr. Ramchand is an honorary visiting scientist to European Nanotechnology Consortium at University of Newcastle, UK. He is also an adjunct faculty, visiting scientist, and PhD guide at Swinburne University of Technology, Australia, along with Professor Ajay Kapoor. Dr. Ramchand is a PhD in biochemistry from MS University, Baroda. He also served as assistant professor from 1982 to 84 at MS University. He has also obtained his MPhil from the same university. He did his bachelor's degree in chemistry at SD College, LLP from 1972 to 1975. He holds a master's degree in chemistry from the University of Kerala. He has also won the Young Scientist Award from Charing Cross and Westminster Medical School, UK for 3 times for the work in the role of membrane pathology in schizophrenia and the development of EPA as therapy of schizophrenia. He has extensive experience in basic research activities, having handled prestigious projects in the US and UK, both universities and in pharma industry. Dr. C. N. Ramchand is the recipient of Kerala Academy of Science Honorary Fellowship. In the last 35 years of 18 top scientists only received this award, and very first from the industry. He has over 25 years of rich experience in the areas of teaching, basic research, drug discovery program, and disease mechanisms, guiding MSc and PhD students, and setting up research facilities. So this is all about Dr. Ramchand, and it's over to you, sir.
Good afternoon. Can you hear me? Sometime I have some throat problem. Probably, hopefully, it will become better. Good afternoon. Can you hear me? Good. One request to you. Please don't talk. Somebody's talking. So that is my humble, simple request. Because it will become very difficult for me, and I am going to talk to you something very important. The first time in India, some molecule is developed. So unless you hear properly, and I won't be concentrating too much on my slides. The reason is that insufficient time. And my special thanks to Professor Soma Das for inviting me over here. And I met her husband, Dr. Das, at the convocation which I was giving a convocation address. And I immediately agreed that the reason is that 2 of my scientists is from, not from here, Calcutta. And they told very great about this Adamas University. They told you should go. That's one of the reasons. And thank you, the organizing committee. Thank you, the Chancellor, Vice Chancellor, all the deans, everyone. Thank you very much. What I'm going to do is that, few minutes I will tell about, what are the various startups they have done. Not much time. Then what I will do is that, what I'm going to talk is about the novel antibody, what we have generated. So it's a novel methodology called phage display technology. So you should know what are the other methods. And just before that, do you know what exactly is novel biologics? Probably may not be knowing. NC and everyone know new chemical entity, what exactly is that? So I need to talk about what is NC, what is biologics, how it is developed in 5 minutes, then we can go for the actual presentation. As I mentioned, the first is, I will briefly, if you talk, I will stop at that point. I can tell you, I used to be a teacher. So if you continue to talk, there are two possibilities and then I can stop. So, you can see various companies. These are all startup companies and I myself have started all this about 10 years back. Please, if anyone has any plans to start your own, please do it now when you are young. Not after, almost at the phase of retirement. That's not a very encouraging thing. But let me tell you, we have made everything very successful. That is one of the things. And you all might have heard about the company called Medgeno. If it is not there, please go and check that. I was an advisor for 10 years and a very successful biotech company. Probably, if you see, many times it comes under the top 10 company in India. As far as biotech companies are concerned, it is next generation sequencing for diagnostic pharmacogenomics. Then what I am going to talk to you now is about, the third one, Saxon Life Sciences, how we have developed a novel molecule using a total new technology. Please do understand, I was head then of Sun Pharmaceutical, which is the largest company at that time also. And there is about 26 pharmaceutical companies in India, but hardly anybody is doing now any drug discovery. No new products, no NCs, no biologics. So in this background really we have done that one. And I will tell you the complexity in starting and making it success is not that simple. So that is what I am going to talk. And MagGenome is a technology what I have developed. Magnetic nanoparticle. And what we have, I have got 7 students with PhD, either in Swinburne University or in MSU. Can you hear me? Backside, can you hear me? Can you show me? Oh, good. So MagGenome is a company which I have done. It's actually a translational research from university. So everybody was working in university and we have used magnetic nanoparticle, magnetic nanoparticle and its use. Magnetic nanoparticle means it is Fe3O4 and you can make it whatever the size you want. It is magnetizable. And we have about 70-80 various products under this company and we have just recently became break-even and we have a facility in US also. And please remember during COVID time, if you remember, earlier you were paying about 2,000 to 3,000 rupees, isn't it? Major cost is there. If you see the collection is there, then extraction is there, then RT-PCR, isn't it? The most expensive part was the extraction. So what we have done is that a novel extraction, patented technology, and the cost brought down. Of course, it took 8 months to 9 months to get the approval from National Institute of Immunology and National Institute of Virology and the ICMR, etc. But we got the approval, and in 9 months we made about 100 crore sales. That's the time the cost of the RT-PCR or COVID detection came down. We have various very interesting products are there. And Teragen Biologics is another company which we are doing for diabetic retinopathy. So both these similar molecule but very different molecule. I will be talking about that also. I also will briefly tell about, we have also introduced a company called Biotech Biopharma Skill Enhancement Program. The reason is that we were not getting the right people to do, especially a startup. There's a big difference. Somebody is working a company like Sun Pharma or large company And a startup, the learning process is extraordinarily high because one person initially will have to do many things. And if you get an opportunity to go and work in a startup, please do that. Don't think about anything, but please also remember 99%, this is the statistics, 99% of the startup in India and 97% in the US will fail product-based, novel products. But that's unfortunately that is the case. And if you see, you also might be knowing that how many drugs India has produced. Anybody can tell? Anyone, any novel biologist or MC, can anybody tell? No, nothing. India has not produced any drug. India has not done any phase 1. Only 7-8 countries in the world could produce novel drugs. What we are doing is generic molecules, or already the patent is over, that will be developed in India. That's what we are doing. But many people tried initially, because when I was in Sun-Pharma also several molecules we are doing, but it is extremely difficult. And in this background, what we have tried to do this one. So what we found, one of the major problem was that it was very difficult to get right people. And it was very easy to do interview also. What we used to do? We'll ask very simple question. How will you make 0.1 molar phosphate buffer pH 7? Hardly anybody, if anybody can answer, please get up and answer. How 0.1 molar phosphate buffer? Don't tell that, you go to internet, you will get the answer. So much of Na2HPO4, but otherwise can you tell? And a large number can't tell how 5% glucose is made. Weight by volume, 5% weight by volume glucose solution. And you also ask which is the simplest form of amino acid. And sometimes I ask how many amino acids are there. We also get answers like 20 to 25 also. I don't know this, which is the 22nd one or 25th one. Anyway, another question is that, why there is a unique amino acid, the 20 amino acid. If anybody can tell, please get up and tell. So these type of very simple questions only we tell. I also ask sometime, because everyone studies carbohydrate chemistry, metabolism, protein chemistry, metabolism, etc. One of the earliest things they study is that about carbohydrate. What is alpha D plus glucopyranoside? What is alpha? What is D? What is plus? What is that pyranoside? Still nobody has to answer. So we thought that it's a, but if you go for an IIT entrance test, these are the type of questions they ask. Very simple. Not what you have done. Very simple thing. So we thought that, we should train them, and when we were training, we were totally focusing on these type of very basic things. And probably IUPAC naming system, configuration, confirmation analysis, and those type of things. Very simple chemistry. Then, enzymology and things like that. For example, no one could answer like, enzyme kinetics part. So we thought that these are some of the things, and that became very successful. About every year we used to take 15 students, and most of them used to get into CSIR, that exam, or get into very IIC, etc. But it was a 6 months course, but intense course. So these were some of the things what we were doing, and I will now tell about. So now basics of drug discovery. Next 5 slides, what I will do is that I'll brief you. Do you know how many diseases are there? Humans have about 36,000 diseases. You all know that we have about 30,000 genes. Now, another thing is that we have about 26 pharmaceutical companies in India, and every day something or the other, and some will be failing. But these are all generic companies. Either it will be that, API company, that means the basic drug manufacturing company, or formulation. But 90% of the companies are formulations. And the maximum what we are doing is ANDA, abbreviated new drug application. You are all from pharma background, you might be knowing what exactly is ANDA. Or maximum is a drug delivery system, what Dr. Wong was telling, similar thing. That is the highest thing many of these companies are doing. In biologics, if you are telling, that is called biosimilars. I hope you know what is biosimilar, what is novel biologics. So I am going to talk about novel biologics. Now, what I want to tell is that, if you really see, there are 2 different types of molecule. One is NCE, new chemical entity, which which is otherwise called small molecules, synthetic molecule. The other one is the biologics. Now both has got different ways of the discovery part. Now two discoveries are there. For example, MC, first discovery is the target discovery. What is targets? Functional protein. What are the functional proteins? Enzymes, receptors, ionic channel, transduction protein, transport protein. Right now, every drug is based on these functional proteins. Do you know how many targets has been identified? I told there are 36,000 human diseases. The first thing is that, do you have any idea how many diseases? We have a very good idea. We do not have not more than few hundred. And I will tell you 25,000 diseases, including rare diseases, we have no idea. For example, we have extremely good idea. We are extremely good in developing antibiotics. Well, infections can be avoided. We are now good in developing vaccines. Many of the viral diseases and other conditions can be stopped. Allergy, asthma, inflammation. Drugs are there, good drugs are there, or reasonably good drugs are there. And certain lifestyle diseases like diabetes reasonably can be controlled. Now, what about many other conditions like, for example, cancer or neurological disease, schizophrenia, depression, mania, manic depression, Alzheimer's disease. In last 30-40 years, 4 molecules for Alzheimer's have come. It's a serious problem. But unfortunately, everything failed, and there is nothing now. No neurological condition really target-based drugs are not there. Cardiovascular disease, very interesting. Please go and read. Now, cholesterol is not connected. Sodium, high consumption of sodium is connected to blood pressure. That is not right. So only few things have been connected. That means type 2 diabetes connected with the cardiovascular disease. But the fundamental cause of cardiovascular disease still do not know. The reason is that those who die of heart attack, there is no correlation clearly not established, whether it is cholesterol or triglyceride or their diabetes, etc. But certain things are not. Those who are walking or those who are doing reasonable exercise, there is a good correlation is established. That means chances of getting a cardiovascular disease is statistically significant compared to the other people. Anyway, so these are the things. What I wanted to tell you is that about small molecules, each target, and only 200 targets are so far known, probably expected for every disease to control. We need to about 2,000 targets are there, which is going on. Where is this target identification research is going on? Anyone has any idea? This targets identification will not be done by a company. A pharma company can't do that. If you do that one, because it is very risky, so that's why in the US, Europe, the maximum amount of money is spent, first of all, studying the mechanism of a disease, finally identifying the novel target, but now there is a much better next generation sequencing has come. There is something called genotype phenotype analysis and transcriptomics, upregulation or downregulation of transcriptomics. These type of methods are there. Anyway, I am not going about that. So what I want to tell you is, as far as small molecules are concerned, the target identification first part, and the novel targets are not. The next part is molecules, MC. There are multiple ways are there. These are normally done by medicinal chemists. Then there is other methods like combinatorial synthesis, and sometimes earlier natural products extracted, purified, used to use. But in the last few years, only 6-7 novel small molecules have come. And people realize that at the peak, earlier 100 years back, the drug discovery was serendipity. Serendipity means accidental finding, which for an example is penicillin as an example, it's a serendipitous. Then subsequently rational drug design and development has come. But unfortunately last few years even small molecules are coming down. That's why everybody is shifted to biologics. Now what is biologics? The discovery and development, whether it is target, whether it is molecule, very, very different. Now I mentioned the targets of small molecules, MC is the functional protein which I mentioned. On the other hand, biologics it is different. For example, our target is VEGF, vascular endothelial growth factor. It is not embedded or based at some receptor or something. It is a moving around molecule. All the cytokines, TNF-alpha, tissue necrosis factor, IL-6, IL-17, IL-2. So these are the proteins which is the target. Now that target identification is a big research program. Again, that is done by universities. Now, as far as the drugs are concerned, these are biologics. Initially, it is the natural, for example, interferon alpha, interferon gamma, those type of molecules. Now, mostly it is concentrating on antibody, which you know, antibody, how an antigen will be injected into animals. You get antibody, purify it, but that is not the way. An antibody, therapeutic antibody has to be generated, which I will be talking to you. Anyway, so what we have done is that how this novel antibody is generated. So I just want you to understand all about drug discovery. In 5 minutes, this is the nutshell. There is a big difference between NCE and biologics, and the discovery part and development part also is very different. And earlier when I was in Sun Pharma, mainly involved in NCE development and some vaccines and other things. Now we are developing totally biologics. And yeah, that's what I'm going to talk to you. I'm not going to tell you, what are the different types of drug, which you know. So this is another, normally a drug discovery in NCE, whether it is biologics, takes about 10 to 12 years time. As I mentioned, discovery part is one. Discovery of not the target, discovery of the molecule. Normally, an NCE development, about 10,000 molecules has to be done, average. That's what. When I was in Sun Pharma, we were making about few thousand molecules to get an appropriate right molecule. And one of our molecules straight went up to phase 2, but failed. Anyway, I'm not going to explain, because if I have to explain this, it will take about 2 to 3 hours. So this also I mentioned, what exactly is that the NCEs or biologics. Okay, now I will tell about the molecule itself. So the company's name is Saxin. The reason why we have put Saxin is that you might be knowing it's not easy to get a name whatever you want. So finally we thought, we'll take from Sanskrit a word. Saxin means, vision. That is what it is. Finally we got that name. And we started the company in Chennai. We have a collaboration in University of Toronto for fast display. That's what we have done. And 2014 we have developed. Initially we had about 25 people. That increased up to 60 people. Now it is very less because we are going for clinical trial. Now Saxin has become Saxin INC. It's a Philadelphia-based company for clinical evaluation, phase 1. And actually it is like a big story to tell why in India it is very difficult to develop drug, why we have to go to US. There is no time. So I will be talking to you about macular degeneration, then platforms of antibody generation, because at least what are the ways people have done antibody generation, you should know. Then, structure based. You might be, you all might be knowing what is QSAR. Your pharma people, please at least keep your hands up. I will otherwise assume that you don't know.
Sir, repeat the question, please.
Somebody has answered.
I asked what you asked about QSR. I couldn't answer.
I don't mind going for an interactive session. That will be.
Sorry,
that is
sir.
will-
I was saying that what you said about QSR, could you please repeat? I couldn't understand.
No, I was asking a question. Can you tell what is QSAR?
Quantitative structure.
Very good. Yeah, it is structure, activity. You need to know the structure. You need to understand the activity. What is the receptor? You have to have multiple different assays, and using QSAR or quantitative structure activity relationship, you improve the molecule. Once you have got the pharmacophore, the next part is that in biology, when you are developing biologics, it is something like that. In biologics, the difference is that In NCA, small molecule, which is roughly about 200 or 250 to 600 is the molecular weight, and it has a particular conformation, and it has to go and bind to a, I think the sound is coming from outside, okay. So this molecule will go and bind in a particular conformation or particular way. And by understanding the conformation of the protein and conformation of the molecule, a medicinal chemist will be able to alter. There are two ways are there by understanding that structures, both the structures, because normally a molecule like, small molecule, how it has to interact, there is 4 or 5 different pockets will be there. Pockets for binding at the protein and the pockets like hydroxyl group, amino group, hydrophobic group, carboxyl group, all these things like different part will be there. And it has to like 3 or 4 different places it has to bind. And what I want to tell you is that using structure activity relationship study you can do that one. Apart from that one, in vitro experiments are there. The same way in biologics also structure activity relationships are little different because it is a protein-protein interaction. In protein-protein interaction, that is, can you close that door? In protein-protein interaction, it is slightly different. So these are our companies based at various places. The third one is one of our labs, and these are our facility. One is at Tyson Park, and one is in Donnelly Center, which is in Toronto, and that is MedGenome's office. We also, MedGenome has an office in US. So these are the, okay, these are, when you are starting any new company, it is better to have advisors. And if you are developing a product, please bring advisors who has real experience in that one. And the third person is Dr. Jayakrishna Ambati. He is the most famous ophthalmologist in the world. If you go and check, his brother Balapurli Ambati is the one who got at 15 years MD from Harvard Medical School. So he is our advisor. Now he himself is developing few molecules. I am also helping him. This is for diabetic macular edema, diabetic retinopathy, some geographical atrophy, this type of few molecules which is reached already phase 1 and phase 2. We are trying to do in India. So these are the other our advisors. Now what is wet AMD? You can just see what exactly. So you can see a normal retina, and before that somebody who has a wet AMD problem, age-related macular degeneration. What is age-related macular degeneration? There is something called macular degeneration, or it can be also called dry eyes. And normally many people, and no one knows what is the causative factor. Normally about 15% of the people who get dry AMD will become wet AMD. Dry AMD is normal, you can have vision, there is no vision problem, but in wet AMD, what is happening is that, after some time, there is a neovascularization takes place. Neovascularization means vasculature. The blood vessel grows. This is because of the upregulation of VEGF. VEGF is involved in neovascularization, vasculature, which is all over the body, and there is multiple types of VEGF is there. VEGF A, VEGF B, VEGF 161, VEGF 125, about multiple subtypes are there. So in the eye mainly it is VEGF 125 and 161. Now what is happening, you can see left side, the one which is showing round, this is the normal retina. The retina has got a keen vision side, macula and fovea. This is the place all these neurons are centered around. Now as long as everything is fine, you will be having a very good vision. Now, unfortunately, some people, which is genetic in nature, their VEGF goes up. What is the role of VEGF? VEGF is involved in neovascularization, lot of vasculature. So you can see right hand side. Initially you won't know. It will be very difficult to find out. Suddenly what will happen is that, after growing like that, it breaks. Once it breaks, you can see that, that left hand side. So, that blood will ooze out and go to the retina and you will lose the vision. So initially what you are seeing is that whatever I have written there, you will be having less of vision, and eventually what will happen is that it will increase and you won't be able to see. So this is the basis of the disease. Now there are drugs are there, various drugs, because one of the things is that photodynamic therapy, laser photocoagulation, not that effective. Then a molecule called Avastin. So just for you to understand, Avastin was never developed for macular degeneration, not for wet AMD. It was an anti-cancer drug. Why you need a something to sequestrate VEGF? Because one of the major problems with cancer is that growth of blood vessel. If you have a peripheral carcinoma, lot of blood vessel come. If you block that one, the nutrient will not go. If nutrient will not go, the cancer can come down. So Avastin was developed and is very successful adjuvant therapy. Now what is happening is that this same Avastin all over the world, not licensed. In India, including All India Institute of Medical Sciences, what they are doing is that they take the anti-cancer drug, which is Avastin. The reason is that it is very inexpensive. The actual drug, which is Lucentis, the second one, which is licensed about a few years back, is very expensive. The started with one injection is $25,000, which is huge amount in India, but now it is about $2,000. Normally about 8 injections are required. That's why, that is one of the reasons we thought that can we make this molecule for $100 in India. So that was the background, we made that one. So Avastin still people are using. It is called off license. No, it is already off patent, but it is not licensed to do that one. But a special permission can be taken, and many doctors do that. It's an approved drug for cancer, but doctors are allowed to inject that one. Then few other molecules are also there. So most successful molecule is Lucentis. Lucentis value in the US alone is $8 billion sales. 8 billion. That means the company's valuation roughly about 120 billion. Now imagine, does India has got any, all our companies together, add up together, because of IPR value is not there, will not come that much. That is the difference when you are developing a novel molecule which is patent protected and which can be used world over. The moment that becomes generic, in overnight the value will come down. Now, this is the whole thing. This is what I am going to, step by step, going to explain to you. So, you can also see, so there is something called drug discovery phase. That is the time we have developed this molecule, the antibody. Now, what is the type of antibody I need to explain? This is not full antibody, it's a part of the antibody. And I hope you know what is the molecular weight of an antibody? IgG, IgM, IgE. Anyone tell just like that? We can just get up. No one knows. Okay. How many amino acids are there in an antibody? No. Okay, 1500 is the amino acid, roughly 148 kDa. And if you see an antibody is Y-shaped, isn't it? And in the Y shape there is a constant region down, the other places are the antigen binding. So you don't require the entire antibody. So what we have done is that cloned express only one part where the antigen can bind. This is what we have done. So our first part was discovering the molecule. And that we have used Fast display, which I have mentioned. Then after that, once you have identified the molecule, identified the sequence of the DNA, then cloning is there. Then once cloning is there, you will have to express. The third part will be, you will have to have a proof of concept. What is proof of concept? You normally need to have an animal proof of concept. In the animal, it is working. So an animal model has to be developed, and that is what we have done. And subsequently, once that is, see, I will tell you, if anybody wants to talk, please go outside. Because it is coming, and because I was talking to Dr. Wong, he also found it difficult to talk. Am I right? Yeah. Now what I am telling is that this is not an organized talk I am giving. The reason is that many things will come from here, so I will lose my concentration. So if you are talking, I don't have any issues, you can go outside and talk and come back. So the 1, 2, 3, the fourth part is that we will have to do the complete process development. It's a very complex process, that getting the approval. Now the regulatory approval, unbelievably complex, and in the last slide I will be showing what are the different approvals you have to take. And let me tell you, we are the only one in India till date got a Nobel Biologics, what is called the C6 approval. Not even many other companies in between, they try, they go back to US or something. So we could get it from India itself. So after that, yeah. So then multiple other studies we have done, ERG study, then regulatory approval, CGMP material generation, investigational IST, then regulatory approvals. Can you see all these things? I can't see properly, that's right. Okay. Then finally, the phase 1 clinical trial. Now, what are the challenges we have faced? So, getting finally the approval C6 approval means it's about 25 to 30 various approvals are required. Anyone who is involved in a recombinant protein development will know. And it is nowhere in the world is there this much approvals also. When you are going to other countries, there are many approvals, but not so many are there. The major problem we face is that, the material, whatever I was telling that we have done in our laboratory, we have the complete facility. But before going to the human, you need to produce that in a CGMP condition. Have you heard GLP, GMP, GCP? Somebody, yeah, very good. Somebody told yes, good. So you have to develop this molecule under GMP condition. Another good thing is that in India and US is the only two places you can do this tox studies with laboratory developed product. But nowhere else it is possible. Everywhere you have to do the CGMP materials is required. The other one good thing is that India also allow, it is not required to do a GLP tox study, but no one will take that risk. That's also there in the US, but everywhere, otherwise everything will be GLP, GMP, GCP has to be there. The other thing is that, which I have not mentioned, it's so important before the data is published or going with other people, we will have to file the patent. This is an international molecule. Now everyone knows that, provisional patent is very easy, but it took almost 6 years to issue the patent. So we filed the patent, and that is very costly. It's not filing a provisional patent. The third phase is the execution phase. Each country you have to pay about $20,000 to $30,000 for filing or the fees. So probably we have spent about $1.5 million. The complete issuance of the patent, including North America, India, China, Europe, Russia, Australia, we have about 8 or 10, which covers about 80% of the market. So I will just briefly tell so you need to understand what is phage display, why you have to do phage display, and what is the methods of generating antibodies. Do you know what is hybridoma technology? At least somebody tell yes. So yeah. Okay. So classically it was a hybridoma technology. You can see B cells. How B cells? You have to inject the antigen. Of course, it's not just an antigen injection. There are ways by which then you have to isolate the B cell. Then B cells will be, can be cloned, combined with the myeloma cells, cancerous cells. Even human cancer cells can be combined together. Advantage is that there is some specific mediums are there, which this group. Advantage is that all these antibody which is generated in the mouse can be transfected and it is immortal cell. That means there is no specific number of cycles. Normally most of the cells, there are QX cells only 7 cycles, or many other cells maybe 20, 30 cycles. So this can grow continuously. And there is a way by which after growing in a medium called HAT, you can select by ELISA the clones and you will get the monoclonal antibody. But the problem is that there is no therapeutic antibody generated using hybridoma technology. It can be used for only laboratory purpose. There are a couple of reasons. You can never make this molecule extraordinarily affinity because the molecule what we have developed works with micrograms inject. But because of the affinity, absolutely affinity matured what we have developed. Even Lucent is very high affinity. If your affinity is low, you can't inject gram quantity into the body. That is one thing. So the second thing is that there is something called immunogenicity. Any foreign molecule, this is luckily not there for small molecule because of organic molecule. These are all protein. The moment the sequence is not connected to your body, the body will produce antibody. That is the problem with if you develop an antibody using hybridoma technology. Chimeric. Chimeric means two different animals combined together, or that is what exactly it means. But in this sense, chimeric, what you do is that you develop the antibody, you take the RNA, mRNA from the animal, because you can't inject to human, isn't it? If that could have been done, an unknown antigen can be injected into human and take the B cells, could have been great, but that is not allowed. So you have to inject into animal, get the B cells, and then what you do is that this antibody has got two parts. Like one is CDR, complementarity determining region. The other one is the other sequences, base sequence, which is not involved in any kind of interaction with the antibody. So that part will be taken from human. And this can be very, if you know recombinant DNA technology, these are all now not at all complex. Extremely, because we do these type of things very, very regularly. The second part is that, what you do is that you completely take human antibody, that is mRNA. What you do is that delete the CDR. So if you see 1,500 amino acids, roughly about 10%, 150 amino acids, roughly about 90 will be involved in CDRs. But about 150 amino acids you remove and you clone and put the hypervariable region, that is CDR, into that, then you can get about 90% humanized antibody. 90% humanized, the immunogenicity is extremely low. But any antibody, therapeutic antibody, if you inject, it is still going to produce some antibody. So that is one of the tests we have to do. And certain amount of antibody production in the body will be allowed. Otherwise, what will happen? The completely antibody is produced, you inject the second time, there will not be any antibody. Now, what is phage? Have you heard something called phage display? What is phage? Virus, isn't it? Okay, good. What is virus? Virus normally having DNA virus, single strand DNA virus, RNA virus, various things are there. This here it is used single strand DNA virus. You can have 30 to 200 different DNAs are there. And if you take a virus, there is something called coat protein. Coat protein is the protein which is produced by this DNA, which is called coat protein, and some of these proteins will be outside. This is what, like for example, a virus has to transport into your cells. That's why if you really know, there are 3.5 crore, 35 or 335 million viruses are there running around. But we have to worry about only 200 virus for our cold, few here and there virus for our disease. What is the reason? The reason is that they are made in such a way that they will not interact with our receptors in the cell surface. If that interacts, that's what happened with corona. It was originally for an animal virus, but then some mutation happened. Of course, the spontaneous mutation happens like that for this thing. We don't know what is going to happen in the future. So that is what happened with corona. So this surface protein, the surface which is protruded outside, will be one of those protein will be used to clone whatever is the protein of our interest. I'm just telling all this as simple as possible. I can't tell anything even simpler than this. But if you have any question, it does not matter. I will finish my talk in time. But if you have a doubt, just ask and I can just explain to you. So that is what it is being done. And this virus will be done using something called phage-mid. Plasmid, you know, the same way. Those infect the phages called phage-mid. And this phage-mid will combine with the protein in the virus and start expressing. So normally this virus will be cloned in such a way that 10 raised to 12 to 10 raised to 13 such diverse antibody, that means our antibody RNA is producing and which will produce the antibody. So this is the fundamental basic technique of fast display technology. Then finally what will happen is that you can see here, in the case of VEGF, this is immobilized antigen. In this case, immobilized antigen is VEGF. That VEGF is immobilized, and any of this virus which produces VEGF antibody will bind. That we will take out and proliferate. This is the fundamental technique of phage display technology. And we have to make name library. How name library is made in phage display, that is what George is going to explain that to us. Now, why this is called synthetic antibody? Have you heard something called synthetic chemistry? You know synthetic chemistry. Any organic molecules can be produced by, I am an organic chemist. So I know anything can be produced if you know the chemistry. Most of the molecule can be done. Why this is called synthetic biology? Now, as per synthetic biology, you can prepare peptide synthesis, you can produce protein, you can produce fatty acids, you can produce lipids, you can also produce carbohydrates. So that means any molecules can be produced. Now this comes under synthetic biology. The reason is that initially we are cloning, then expressing, and whatever that produces that protein is called using synthetic biology. Though it is kind of recombinant DNA technology. Now this is VEGF. Have you heard of something called VEGF, vascular endothelial growth factor? You heard about that. It is mainly, it is a transduction protein which is involved in many situations. Like, it is kind of, cancer can be produced that if it is uncontrolled production is there. So VEGF is roughly about 500 amino acid. It is anti-parallel. You can see the red one is in one direction, the other one is by the other direction, and which is linked together. This is basically the conformation means three-dimensional conformation of this protein. Now, both these sites, because both these sites are identical, both these sites, the VEGF antibody can go and bind. So this is fundamentally the principle. So this is the molecule. I am not going to explain anything more because no time. I started at 3:15. I think I have 10-15 more minutes. Okay, now this is the way. For example, when they made Lucentis, from there up to here, Lucentis. Now, how they made Lucentis? Lucentis was made. There was no fast display technology. What they did, they have taken VEGF and you have to inject into mice or rabbit, normally mice. Then you take spleen, what is called splenocytes, take the B cells, and as mentioned earlier, finally the antibody will be produced. But these antibody, now the complexity comes. You got an antibody which binds to VEGF. But the problem is that that affinity will be very, very, very small. It might be having affinity. In order to get an absolute affinity, that is the way. What we have to do is that you have to mutate continuously. The way which it is done is that one is you take the structure of that and you take the structure and you express the protein, bind it, and using molecular biology you will be able to tell how is it binding, what is it binding, which part it is binding. Now normally, in order to get a very good binding, there will be 4 different or 4 to 7 binding pockets or touching should be there. Paratope and epitope, which you might have heard of it. So there should be an interaction, and these interactions should be below 4 angstroms. If it is above 4 angstrom, there is no interaction. If it is below 4 angstrom, there is interaction. If it is 3 angstrom, it is very strong. Suppose it touches at less than 3 or 2, then it is irreversible binding. That also is not good. So that is the way they have done. What is called site-directed mutagenesis. That means identifying all those things, then mutate continuously various parts, and finally they go to Lucentis molecule. And the advantage is that by all these doing, the company became very, of course it is Genentech. And in US alone the sales was about $8 billion. $8 billion is 70,000 crore, which the top 3 company combined together will not even have, each company has got 700 to 800 products. So please do understand, this is one country and the valuation of that product is about 120 billion. Valuation means if I want to go and buy that molecule for myself to sell in US, that is the price I have to pay. But what we have done is that the basis of the structure we have taken, and using fast display we have developed FAB201. We have developed 2 molecules, FAB201 and 293. The second molecule, we have all decided it is Ramsumab. My name was put. In the morning someone was telling, Bevacizumab, I don't know what is the basis, so they put RAM, Sumab. So once it comes out you can see, but it will be always known by a trade name, not this. I can't even pronounce that name, let me tell you. Okay, the other thing is that, one interesting thing is that we have developed everything and we were almost ready to get the approval, that what is called the C3 approval. C3 approval is very important. After that toxicity goes and finally C6 approval. Multiple things are there. C3 means C4 approval. Now the regulators, so many times I went there, every time you go there, there will be so many questions. I am very sorry to tell, but those who are sitting is about 36-40 people, all from academic background. They have never done the actual molecule. The difference in US, because we submitted to US, it was very different. In 30 days I had very clear-cut 3-4 answers came. And that is what we have to answer. Anyway, now paratope epitope binding, they told us that, how you know this is novel molecule? Please tell that whether our antibody, FAB201, is the same amino acids or same epitope it is binding. Then we had to prove that. Where VEGF-R1 is the receptor which VEGF goes at, VEGF-R2, and where this VEGF, what are the pockets, what are the amino acids which goes and binds, and we have proved that the one which is Lucentis is different, ours is different. What you are seeing is that the pocket where it binds. So which I mentioned what exactly is the structure-based drug design. Basically, identify the ligands, correct binding geometry in the binding site, predict the binding affinity between the entities, mutate the residues and create new binding site, evaluate the change in binding affinity, predict new interactions that can have better binding kinetics. So these are the various interactions, and I know that it's a very complex thing. If anybody has got any doubts or something, you can always ask that one. So these are the various interactions: electrostatic, electrodynamic forces, steric forces, solvent-related forces like hydrogen bonding, etc. This is basically the structure of Avastin. So this is VEGF dimer, which binds, and the top and bottom, which I mentioned. If this actually rotates. If this is VEGF, this is one binding, this is binding, this is Avastin antibody. Yeah, this is again the interactions with Avastin, and which I mentioned that the interaction should be less than 3 angstrom. Yeah, again this interaction only. This is VEGF dimer, again this is Lucentis and the Lucentis interaction, the same. This is the molecule what we have, Fab 201 and Fab 293. 1 minute, I don't know. Okay, so this is exactly what we have developed, 2 molecules. Now this is the process of development. This is the cloning. Few things I will explain here. So after cloning, there are many things. This is a 3-hour talk, so I'm just completing everything in this thing in about another 5 minutes. So, this is the bacterial expression clone. What we have developed, the whole thing is in bacteria. What you are seeing is the clone. One important thing is that, we have used elements of 4 inducible expression. There are various products are induced. That means at particular time, the plasmid will start inducing or producing your protein. We have used 4. That means, normally something concentration goes up, the production takes place. Here it is not like that. When we start the fermentation, there will be high levels of phosphate is there. When the phosphate comes almost at the level of 0, the production will start. So this is a new type of inducer. Now these are about 25-30 different clones we have developed, and everything went through, and finally we got 2 different ones. Both the ones we are using, one for diabetic retinopathy and one is for macular degeneration. So these are the various assays we have done. There is one called quantikine assay, mainly for EC50 determination. SPR, surface plasmon resonance, which we went to All India Institute of Medical Sciences to do, because SPR, because we have to do only few assays and it costs about 3 to 4 crore. Then in vitro biological assay, and this is done outside, just to make sure that, and you can see the Fab 201 is the most potent of all these molecules. Then in vivo assay, we have also proved that in vivo, that means in animal model, the molecule was far superior than even the best of the best. So by this we have found that we found ED50 value, because when you are going for clinical evaluation, one requirement is that even if you are going for small molecule also, ED50 to understand what is the starting dose to be given to human. In order to do that, and what we have found is that ED50 is about 13.6 microgram. Probably we can use something like 25 to 30 or 40 as starting. So this is the upstream process. I'm not going to explain. Upstream is that basically after the cloning and other thing, all this comes under upstream. That means cloning part, expression part, laboratory scale up, then goes to the ferment mills. and after the fermentation there is a downstream. You have pharmaceutical biotechnology here, is there? So you might have studied all these things. So in downstream processing, you have to do the purification. So since it is an antibody, the purification is comparatively simpler compared to other molecules, because you can always use other ligands to purify. It's a 5-step purification process. And so these are some of the data which we have to give to RCG. Upstream scale-up growth profile, downstream scale-up chromatograms, STSP profile. Now, another interesting thing I will tell you, we have used first Lucentis because this is something similar to Lucentis. They have published, they have patented also. So this molecule, our formulation, we exactly used. What is happening for every protein immediately after purification. Either it has to go into the freezer, otherwise you have to formulate. So what we have done is that we have formulated using exactly similar to Lucentis. It did not work immediately within 2 days. But we changed, that means whatever they published. So I just wanted to tell, it's not necessary that something which you are seeing in patents or papers need to be right, especially because sometimes very critical thing that they can withhold. And you can also see the stability of the molecule after our formulation over a period of, now it is 3.5 years, absolutely no change at 4 degree, and we have also found that this is stable even at 30 degree and 37 degree for 2 to 3 months. And what you are seeing is that reverse phase HPLC, and what we have done is that over several days apart we have done, there is no change. This is size exclusion chromatography. So these are the other things what we have doing, that we have to check the isoelectric point, then we have to do intact mass, then primary structure. This is a complete sequences. Then something called CD spectrum, circular dichroism, because there is no changes in the conformation end to end, and this is a disulfide bond. Another most important thing what we have to check is that it is if you see the molecules, something which has to take orally, if you have to put it to the skin, less of stringent regulatory requirement. If you are taking orally, it becomes more. If you are injecting, more. If you are injecting IV, it is more. And something if you have to inject into the eye, it is the most stringent requirement, because we have to have kind of endotoxin absolutely low. Imagine this is coming from bacteria. Now we will have to completely remove that one. Then, one of the ligand or one of the affinity chromatography we are using is protein L, and we have to make sure that protein L is not leached out. Host cell protein. Host cell protein. Host cell means bacteria. No bacterial protein. Because we don't know a similar protein with a similar molecular weight, along with that comes out, we won't be able to detect. So host cell protein, and how we have done is that host cell protein, these host cells are taken, broken down, injected into rapid, antisera is taken, and the same antisera has to be used. Then ELISA method is developed, and the same one has to be used continuously, even during the production period. Then another one is host cell DNA, and any host cell, that is bacterial DNA, should not be there, and sterility. When we went to US for this one, they told that one of the most important thing is that the molecule has to be absolutely sterile. And you can see that, product related impurity, absolutely nothing can be seen. So this is, and Lucentis head-to-head comparison, you can see. So Lucentis is currently and was the best molecule, and we see, another thing is that if you are developing a molecule, there is something like superior molecule, non-inferior and inferior. If it is inferior, they will never give approval, Because I know many people are developing several molecules, formulation. If it is an inferior to, have you seen another paracetamol? It is not there. I don't think anybody will develop because it has to be better than the existing paracetamol. Otherwise, it will not. Any molecule it is like that. Because if you see anti-allergic, many first generation, second generation, third generation, there are some possibilities to you. The same is the case here also. If it is superior, great. And if you are not developing a superior molecule, no chance for the commercial success. If you are making a non-inferior, that is something similar to Lucentis, people will think, why are you going to buy this one? Or the doctors will think, because already Lucentis is there. So anybody who is involved in drug discovery development, you always have, if you are developing an existing molecule and competing with that one, you have to make something better than that. Okay, this is the various process what we have done, and this is the regulatory requirement. I'm not going to explain that, but let me tell you, initially when, I think this is the last slide, so when I thought, as a scientist, you always think discovery is the greatest thing. Then, when I started working, especially with Sun Pharma, I realized that development takes such a long time, 5, 6, 7 years, hundreds of experiments, hundreds of assays, standardized, and one thing goes wrong, everything goes wrong. Then I realized that when I went into regulators, that is the most complex thing. But now I also started commercializing our MagGenome product. It's such a good product, and the complexity is so high. So if anyone wants to start a company, first thing what you have to see that how you are going to sell. Is there really going to be a market? Don't start the other way. And this itself is that stories to tell each time, each regulatory approval we have got from there. So I think that is the end of the slides, and yeah, so we have CGMP manufacturing. Another thing, just I will tell you, I mentioned about this CGMP manufacturing. Yeah, I'm only overshot by 2 minutes. So I started at 3:20, now it is 4:20. One thing is that the CGMP manufacturing, I went to Lonza. You must have heard that Lonza has got both bacterial as well as mammalian cell expression. Went to Singapore. They gave me a call for $13.5 million in 4-year time to make. How a small company where everything depends on all these things that immediately we decided. Then a couple of other companies, Indian companies like Biocon gave 3.5 million. Finally, we collaborated with another company, one of my friend's company. He helped us. The same thing we did for 3 months and in $200,000. So that is, I have one thing to tell you to students, more than this conference, see that how you can make the network. If you see many of these people I already made network. I'm going to Guwahati, Dr. Murthy's place. I've invited Dr. Wong, then Dr. Prajapati I have collaborated. So that is what I have done when I went to NIPER also. So those who want to go to a product, so please use this platform as a platform to network. And network can do quite a lot of things. And thank you. I think I'm not going to explain this. This is the final product what we have made. What you can see, the right hand side. So next is investigator supported trial. Yeah, this is some other talks which I have. Thank you very much. Thank you everybody. Thank you.
Thank you, sir. We have one more sessions. So we are in shortage of time, and most of the audiences are from UG and PG backgrounds. So what we have learned from these scientific sessions is extremely good scientific research activities. We have some few keywords actually, or some takeaway that I can explain to the audiences. First, AMD, that is weight age-related macular degenerations. It's a serious concern nowadays actually, which will produce some vision-related activities. Your visions will be reduced. And second TACI is the VEGF, anti-VEGF drugs actually, that is the vascular endothelial growth factors. So anti-VEGF drugs that will be used as a first-line treatment for this AMD. Now we have some more takeaway. For examples, like we have learned from the presentations, there are few drugs. For example, like ranibizumab, aflibercept, brolucizumab, these are the drugs actually which will be injected through intravitreal routes. And there are some synthetic product which will be act as anti-VEGF, which will reduce the growth of abnormal blood vessels in the eye, which is the major concerns and what we have taken from these presentations. And one more thing, this anti-VEGF synthetic product, which is quite correlated with the gene therapy, because nowadays there are few anti-VEGF products, for example, like RGX-314, which is using nowadays for latest example in the treatment of AMD, and that is quite correlated with the AAV, that is the adeno-associated virus. So we can correlate this with the gene-targeted drug delivery also, but we have some questions, only one or two questions I can ask to you, sir. So my first question is, what are the potential safety and side effect concern of using these synthetic fat fragments?
I think that was a question, no?
Yeah.
So there are a couple of things are there. You know, one interesting thing is that this Lucentis being $8,000 per injection, the efficacy is only between 35 to 45%. When I went to Thailand and Singapore, the efficacy is only 35%. In the US, it is about 45%. Now, that's why people are trying, you know, understanding the genotype.
Yeah.
What type of gene? For example, if you are injecting into the eye, your protease enzyme is very active, this can break down. So they are trying to correlate. But then that may not have any, that was one of the reasons what we have developed this molecule, so that, you know, there will not be any inflammatory problem also. One of the reasons is that Lucentis, when injected 500 microgram, there is a lot of inflammation was there. So there was a problem associated with that. They reduced the molecule to 300 microgram. Now, as far as you know, the tox study or the safety is concerned, one thing I can tell you, guarantee, that if you are taking, if you build, I mean, an allopathic drug, that's the only thing you can believe. Allopathic drug, because so much of testing, and that is when it is introduced. For example, when before going into phase 1, so much of tests we have to do, and everything should be accepted by them, even for the first patient, the lowest dose. If there is a single problem, that's the end of the molecule. So as far as safety is concerned, there is no issue. I will tell you, you know, osteoarthritis, the molecule was introduced. Not after Brufen, I forgot the name. It was an excellent molecule. It is a specific COX-2 inhibitor, only cyclooxygen, not COX-1. But what was happened? There was some, all these COX-CYP, multiple COX-CYP, 2 or 3 COX-CYPs, and what was happened? These are all withdrawn now. Celecoxib, Rofecoxib, and one more is there. I don't know which are the one. Maldicoxib. Yeah. Anyway, what I want to tell is that, you know, in the US, during phase 4 or post surveillance, they found out that roughly between 5 to 10 people die in a million, those who are taking. The moment they confirm that the molecule is withdrawn, everywhere. So that safe is allopathic molecule. I don't believe, though I am also working on a lot of natural products, We don't know what we are getting because no testing, nothing is there. It is my personal opinion, nobody is.
And it's a major concern about the costing actually. There are several injections, and per injections it was like 20 to 50,000 cost. Nowadays it has been reduced. So professionalism is required, because if you will continue the treatment procedure without professionalism, you cannot continue. My question is, if you will discontinue the process in between the treatment, suppose you are under the treatment of AMD, so you have to take multiple injections. So per injections, there are a limiting, there are a cost. There are some cost is adamant, means like per injections it will be like 20,000, 50,000. So if any patient will discontinue the treatment, suppose if he has taken 2 injections and if he will discontinue, so what will happen for the next?
Alright, the question is that because so much money is being spent and in between the patient want to discontinue, let me tell you, when you are injecting, which I mentioned, 35 to only 45% is efficacy. In the first or second injection, the doctor will decide whether, okay, this person is not efficacious. The other one is that, you are continuing for 3-4 injections, it is working fine, and if we discontinue, you will lose vision, definitely. And after 1 year, if it is working well, then there will be continuously every 3 or 6 months, again this has to continue. So I don't think if it is working well, no patients will discontinue.
Okay. And my last question is, how does synthetic anti-VEGF fab compare to existing anti-VEGF treatment?
There is no difference whether you have made an antibody by synthetic method, that means fast display, or the classical way.
Okay.
Antibody is antibody, and Lucentis and our molecule, their only CDRs are different. The base structure remains same. The CDR has to be different. That is for your patent purpose and novelty and better efficacy. There won't be. Otherwise, there is no difference.
Thank you so much, sir. So any questions from audience? So thank you so much. Thank you, Advance University,
Sir, I have
for
a
giving
question.
me the.
Not related
Okay.
to this. VG or the fab.
I got.
Hello, sir. I am here. Like,
Okay. Yeah, please.
the very first topic you mentioned was novel drug delivery. My topic isn't on the antibody or the antigen fab, the novel drug delivery you did. I'm talking about the NDDS you talked about at in the very first timeline. In India, there you said that there isn't novel drug delivery has been done, or NDDS not has been worked in here. As you can see, it's an academic place and lots of students are aspirant of R&D. How would you suggest the new generation to focus on the new novel drug?
Yeah. The first thing is that you mentioned NDDS means new drug delivery. That we are doing.
No, I'm sorry, sir.
Novel drug
discovery. I'm sorry.
Novel drug discovery.
I'm sorry, sir.
See, the first, what did I do? I was in academics, I was teaching in UK, and I came and I joined, and I got an opportunity, and now I'm only into that. Now, what you are telling is that, where you can do, what did I do? I started my own. And if you have an idea, you can start your own. Now, the thing is that why this big pharma company is not doing novel drug discovery, but they are doing many other research. Sun Pharma is spending about 5,000 crore per year for research. It's not a small amount, but now they are slowly getting into biologics and other things. So, but let's assume that if you are very interested in novel drug discovery, novel biologic discovery, there are 2 opportunities out there. Why don't you try and go to US or Europe? This is what everyone is doing. That's what my first job was in US. And there is enormous opportunities there. But you can always come back, you can start your own things, or you can join and initiate with them or compel them to do it. So, but though new drug discovery is not too much in India, many other things are there. I assume that is that your question.
So isn't that it is implying that India doesn't have that facility to like follow up the novel drug discovery here? So when we are wanting to like pursue that, we have to go in other country. That you are suggesting.
Correct. See, okay. Your generation is so lucky. And if I tell about my story, what I have studied, I will be amazed. And I am so unlucky, I feel that I did not born in this generation. Because the opportunities are significantly high. So, the only thing you are questioning is that why India is not doing novel drug discovery in India. But please remember that Indian pharmaceutical companies are there. You know, if you see GSK is there, Novartis is there. They also have. Novartis has a very big facility. They are doing. It's not like the entire thing end-to-end they are doing here. Part of that should be there. Lot of clinical trials are happening here. Lot of regulatory submissions are happening. Lot of BA/BE studies are happening here. But whether Indian company, like you know, for example, see, Sun Pharma is now started because one of the molecules has already 2 molecules have kind of reached phase 1, phase 2 in the US from here, developed here. So it is not that absolutely this thing. So what I was telling is that you know you can try in India, but then a lot of opportunities overseas also is there. Overseas means not every country. All these drug discovery is happening only in the US, Canada, Europe, Australia, like that about 10-15 countries are there. No other countries are involved in doing this drug discovery because it is so complex. I don't think anything as complex as drug discovery. Because if we could send a rocket to anywhere we want, but the drug can't be produced because of the uncertainty, tremendous uncertainty. That's why.
Thank you, sir.
Thank you so
Thank you so much. Now I request our chairperson for this session, Dr. Shoropananda Mukherjee, to kindly felicitate our distinguished speaker, Dr. C.N. Ramchand. Thank you, sir. Thank you, sir. Now, I request Professor Dr. Sanjoy Dey, Associate Dean, Department of Pharmaceutical Technology, Adams University, to please come up on the stage and felicitate Dr. Shoropananda Mukherjee for this session. With great respect and admiration, may I now request Dr. C.N. Ramchand to accept his token of appreciation from Dr. Shoropananda Mukherjee. Sir, I request you to please be on the stage. sir. Thank you, sir. Moving forward, we now begin with the scientific session 4 on the topic Solid Lipid Nanoparticles Transforming the Drug Delivery for Better Healthcare. May I now welcome on stage our distinguished speaker, Professor Dr. Bhupendra G. Prajapati, Department of Pharmaceutics and Pharmaceutical Technology, Shri S.K. Patel College, Pharmaceutical Education and Research, Faculty of Pharmacy, Ganapath University, Gujarat, India, and esteemed chairperson for this session, Professor Dr. Himanshu Shekhar Machi, Dean Academics and HOD, JIS University, Kolkata, India, to preside over the session. Sir, please, I request you to be on stage.
Hello. Honorable Vice Chancellor, Adamos University, it's my privilege to be on the stage and introduce the next speaker in front of you. Dr. Bhupendra G. Prajapati is a professor at the Department of Pharmaceutics at Sri S.K. Patel College of Pharmaceutical Education and Research, Ganapath University, North Gujarat. He is also an adjunct professor at the Faculty of Pharmacy, Silpakorn University, Thailand. With over 23 years of academic and industrial experience, he has significantly contributed to the field of pharmaceutical education and research. Dr. Prajapati has received his PhD degree from Hemachandracharya, North Gujarat University in the year 2011. He has graduated and post-graduated from MS University, Baroda in the year 2000 and 2004 respectively. He has received a number of awards and recognition in his 23 years of career. He is AICT's Career Award for Young Teachers in the year 2013. He is a distinguished associate professor taken ex-India 2017 by CSI Mumbai. He received the President's Award for the Staff Excellence in Research, Ganapath University, twice in 2019 and 2021, and has also received the President's Award for the Capacity Building from same university in 2020 and 2022. He has also received MDPI Pharmaceutics 2022 Outstanding Reviewer Award. He has around 200 national and international publications in journals of repute. He has successfully obtained grants from various research projects, staff development programs, seminars, conferences, and travel grants from both national and state government agencies. His industrial consultancy projects guidance have been highly valuable to the Institute, actively involved in the lipid-based drug delivery and nanotechnology formulations. To talk about a little bit of the research contribution, he has got 2 patents granted and 7 number of patents were published or filed to the Indian Patent Office in the field of novel drug delivery system. He has edited 20 number of international books with Elsevier, CRC Press, Wiley, Apple Academic Press, Jenny Stanford, etc. He has contributed over 150 number of book chapters. He has delivered more than 50 expert talks and invited sessions at national and international conferences. He has guided nearly 40 number of PhDs and nearly 60 number of PG researches in the field of nanoparticulate drug delivery and bioavailability enhancement field. He is a fellow of Royal Society of Chemistry, the member of Pharmaceutical Society of Japan, life member of Indian Pharmaceutical Association and Association of Pharmaceutical Teachers in India. I feel privileged to introduce you, sir, on the dais of Adams University. We are eagerly waiting to have your talk on solid lipid nanoparticles transforming drug delivery for better health care. Thank you, sir. The stage is all yours.
Thank you. Thank you. Shubh Sandhya. Hopefully, we have some good interactive session, although it's last one. And I would like to clap for the students who have been sitting over here. It's been a tiring day. See, good listener is always a good learner. And good learner never stops, especially in the research journey. Like we have different sessions starting from the phytopharmaceuticals. We have driven to the nanocarriers, biopolymers, technology part, drawback to the biotechnology, monoclonal antibodies, VEGF. So I'll be going to talk about the solid lipid nanoparticles, and as per my today's understanding, there is a different kind of the students groups are there: pharmacy, biotechnology, maybe basic sciences, applied sciences, along with some food and nutrition fellows are there. So maybe we'll try to club the things or examples along with the basic understanding. Although I have some experience in the pharmaceutical industry, around 2 years in tablet manufacturing, which we all understand. So we'll compare, or we'll try to give the examples compared to the solid oral dosage forms to the solid lipid nanoparticles. So before starting the session, I would like to thank the conference organizers, the team, for tirelessly working from last few weeks and organizing such a impactful event that will be for students and with students. So solid lipid, as we know, it has been well known from the ancient years that many of the natural compounds are taken along with the honey. We know that some our dadimas formulas are there which has been taken with the honey. Some are taken with some oils. Even till date, if we have a ear pain, that has been taken with the clove, and we'll boil in some oil, and we used to have that, make it sterile, and then we can reduce the pain in ear. This is a classical example which we used to have from not many years. Now it has been scientifically proven stepwise that using the lipids can improve the permeation, which is one of the rate limiting step for the absorption of the many of the drugs. Especially like we have a previous session, there has been biotech derived molecules. If you took any of the biotech derived molecules, their molecular weights are too high. So difficult to absorb, they are highly unstable. Although sir has maybe used some words in previous session, that drug is stable for 3 years at 4-degree temperature. But lately he said that immediately it is stable at 25 to 30-degree temperature for 2 to 3 months only. 2 to 3 months. So that's very short time. And if you took it as a stability, a shelf life, it becomes one of the barrier for industrial scale-up. If drug is not stable at all, we could not process, we could not able to sell it in a market on a larger scale. So sensitivity, stability, that is one of the biggest problem with the new molecule which has been arising. Then there are complex processes are there, which makes these structures larger and larger. We combine the one molecule with another, structural enhancement are there. And because of that, the majority of drugs which has been synthesized are having a lipophilicity, which has been creating a hurdle towards the solubilization. And on another part, it has been reducing the dissolution. If drug will be not dissolved, it will not going to absorb from the GIT. So we will be focusing on this part, that how we can improve the drug absorption and permeation. Okay, so in pharma we have the drugs which has been synthesized are classified into the 4 basic class based on the solubility and the permeability. That is well known phenomena by which we can identify the basic physicochemical properties are there, based on which we can identify that what kind of the formulation we can approach for a new drug which has been synthesized. So class 1, highly soluble, highly permeable, there is no issues are there. We can make any kind of the formulations, injectable, solid oral dosage forms, or even any kind of the liquid orals. Topical preparations may have some issues to permeate through for DCS class 1. BCS class 2, there is a solubility issue, so low soluble, high permeable. So if we want to deliver that drug into the human body or maybe to the animal, it requires to be solubilized. To improve the solubility, we have a different concept which we can use, and if we improve the solubility, we will be going to improve the permeability because they are freely or maybe having a good permeability. The BCS class 3, reverse to the class 2, there is a permeability issue out there, but solubility is not a class. We are interested in that. We can improve it by coating them with using some kind of the lipophilic material so that the permeability can be enhanced. Class 4 most difficult, and many of the molecules nowadays synthesized especially for the anti-cancer, that has been of BCS class 4. And even though along with this all, one of the biggest problem is total dose that we are going to deliver. Higher the dose, the challenges will be much more. So if we can improve the permeability, we can reduce the dose also. Permeability, if we are improving, we are reducing the total dose which is required. And the all regulatory guidelines, if you take, they have one motto: producing a safe and most effective therapy. So safety comes first, secondly the efficacy. They should be effective in nature. So zero side effect, ideally we can say that the drug should not have any kind of the side effect at all. And they are effective with the minimum dose, so potent. So if you go through the literature and well-established theories, there are some problems are there with the drug which has been developing day by day. That has been having a low solubility, especially concerning regarding the oral dosage forms. Then low viability, and these two are we can basically take as objective to develop the solid lipid nanoparticles. So for lipid, we know that oils are there which has been fluid in nature. We can convert it to the lipid nanoparticle or nanoemulsion. These are the simplest solutions are there for many of the drugs which can be solubilized in oil. Although they are not solubilized in water, we can develop nanoemulsion, microemulsion, or we can go for the lipid nanoparticle. Then why solids? Why we have to convert it to solid? Because they are more stable. They can be transported, they can be delivered in easy way. We can stabilize them. There is not much change are there. We are using preferably certain lipids which are solid at the normal condition, at a normal temperature. Anything which has been having a solidification above 50, that can be easily used for making the solid lipid nanoparticles. And there are list of benefits are there, that it has been improving the overall encapsulation. So the percentage encapsulations can be up to 80 to 90% also. Even it can be done of both way, matrix as well as the encapsulated form. If if you are encapsulating, it can be used for even the targeted purpose by selecting the lipids which has been specially designed for binding and delivering the drug to the receptor sites. We can improve their overall circulatory time by coating them by using a different kind of the water-like molecules also. They are good chemically stable and overall they can enhance the oral drug delivery. Even these solid lipid nanoparticles, if you are selecting them for the control release or targeted release, it can easily achieve without changing much of the formulation composition. Only the excipient selection will be crucial over that part. Initially the idea was behind the permeation enhancement, but the concept of solid lipid nanoparticle can be extended to the topical and transdermal drug delivery, and it can be extended to the parenteral formulation also. And as Sir has discussed about the macular degeneration, it can be extended to the ocular delivery or even the some of the mucosal drug delivery system. So these are some of the merits are there. A low toxicity that is based on the selection of excipients. If you are selecting the available synthetic or semi-synthetic lipids which has been like our body, which has been creating, ranging from the cholesterol derivatives to the many of the natural oil derivatives like castor oil and the coconut oil. They proven to be safe in even a higher dose. They are green synthesized, not many solvents are required. The cost can be reduced except if you are going for the end product with the lyophilization. The cost can be controlled. Some of these challenges are still there into the producing on a larger scale, like high-end equipments are required. If heat generation are there, they may be going to create a problem. A lipid which should be highly pure as much as possible to reduce any kind of the microbial and the physicochemical changing and loading, that we have to optimize our process appropriately so that it can provide a highest possible loading. Although it's not new, it's around 5 decades where it has started from the producing a micro to a nano lipid formulations, and slowly it has been transited to the solid lipid nanoparticles because it's convenient to deliver, it's more acceptable and can increase the higher entrapment efficiency. So it has been broadly defined as the second generation of the solid lipid nanoparticles. Now, many of you are maybe from the PG or maybe MPharm students are there. If you want to synthesize a laboratory scale solid lipid nanoparticles, maybe the low cost or maybe the available simplest material in your laboratory as a lipid is stearic acid are there. Maybe stearic acid is available. You can have citostyrene alcohol. You can start with these preparations to be used to make a practice to create a solid lipid nanoparticles with appropriate selection of the emulsifier, which allows them to reduce the size, remain in a separate form with high energy mechanism. The energy which has been provided to reduce the size can be done by different way. Either you can go for the high speed homogenizer, so the speed rotor stator design are which has been agitated to produce or reduce the size. You can go with the high pressure homogenizer, which is industrially scaled and accepted technique is there. So HPH technology which you can use. You can also go for the sonification, which requires slightly higher amount of the emulsifiers compared to the other two technique, and may have some ununiform size reduction are there depending on to the sonication time and the speed which you are using. So proper selection of the lipid and the emulsifier which is necessary for making a formulation. You can go for the QBD also, maybe you utilize a factorial design, or maybe the different kind of the QBD designs are there. So overall, we have to make a homogeneous mixture of the solid solution. We have to transit it to the drug enriched cell in matrix to the coated form in form of drug enriched core, where outer side is lipid barrier and inside you can have anything which you want to coat. Now the advantage in this process is you can have both kind of the drug can be utilized. You can use a water soluble as well as the water insoluble drug. It maybe depends on like a liposome. It will remain into the core or into the layer. So either way it will be there into the lipid or either coated into the lipid. So these are the techniques are there which has been used into the lab scale or either into the industrial scale. I started with the high pressure homogenizer because it has been widely explored in the industry, where stepwise you have to process for melting the lipid, solubilizing your drug, then you have to pass it through the one fixed size piston with high pressure. In this process, to stabilize them, we use different kind of the emulsifiers which has been composed from 0.5% to the 2%. And this cycle of passing this melted lipid along with the emulsifier and drug, which is repeated for number of times till we get a good narrow distribution of the particles. So initially, in few cycles, you will get a macromolecule. Then as you proceed for the further cycles, it will reduce the size. Over here, you can change the instrument parameters with the pressure change also. You can gradually start from the 2.5 to 5 K pressure to 20,000 pressure. So as you are increasing the pressure or number of cycles, your reduction in the particle size will be there to the desired number. And this process will produce a good PDI, which is desirable particle size distribution, with the targeted QTPP achieved in a few minutes. It is not lengthy process, but the instrument cost is slightly higher compared to the other laboratory technique. And at the end, as soon as you are cooling down, gradually the lipid will solidify and your nanoparticles are ready for supply. For separation at laboratory scale, you can use different techniques. You can centrifuge and separate them. If you don't want to centrifuge and if you don't use many of the solvents, you can directly use a concentrate to mix with some cryoprotectant and lyophilize them. Or you can mix with some of the adsorbents and spray dry. If you are using solvent, then it is required, otherwise you don't require to go for spray dryer also. The other are the emulsion solidification. You make emulsion, solidify by evaporation, and you'll get a nanocolloids. Over here you may have a uniform size or not, depends on the stirring speed and equipment which you are using. The high shear mixtures are low cost, can be used at a laboratory scale, but the size variety will be too large. So that may create a problem into the drug release. Once we are going for the batch to batch variation, it may create issues. Even in some time, the stabilities may be lower than the HPH. Similarly, we can go for the micro emulsion creation, and then a dispersion will be solidified, converted to the nano formulations. A solvent emulsification evaporation is there, where there is organic solvents are used, where there is in place of melting, we have been dispersing the drug and the lipid into the common solvent, in which the both lipid and drug have a good solubility, and then we are slowly evaporating the organic phase to get the solidification of the lipid. In this process, again, control of the temperature is very much necessary. The temperature should be such that it will solidify the lipids in a controlled manner. It should not be too high or too low that immediate precipitation are there. The another two techniques are solvent diffusion and the solvent injection. These are not much used into the creation of the solid lipid nanoparticles, although they have been for liposomal and endosomal formulations were widely applied. But although we can use it where there is a dilute solutions are there, where there is a very low concentration of the lipids are there, they may be successfully utilized for creation of the lipid nanoparticles. So these are some of the formulations which has been created by using the different techniques. The drug which has been explored are of different class and different lipids which has been listed over here, which has been used by the methods which has been suggested. At an industrial scale up, and preferable method is the HPH, either it can be hot or maybe the cold homogenization, high pressure homogenization is there. These lipid nanoparticles, solid lipid nanoparticles requires to be analyzed whether they have been prepared as per our standard or not. So whatever the targeted quality parameters are there, whether we have achieved that or not. So for that, we have different methods are there which has been applied. On a larger scale, the interests are primarily at the level of the size distribution and the charge, if any, because all these three things are combined together affect the stability and effectiveness. A charge is very important for binding also, if we are preparing a charged solid lipid nanoparticle. Otherwise, that is only required to be differentiate between the two particles to getting aggregate. So that is as far as possible we have to maintain. If we are using anionic surfactant, obviously they are on negative side. If we are using cationic surfactant, they are obviously on a positive side. So that largely depends on to the surfactant which we are using or emulsifier which we are using. The particle size that more or largely depends on to the process parameters which we are using, methods which we have selected, and the dilutions which we use, at what dilutions we are working on. Then we have to go for the surface morphology. Solid or there we go for the sand, or liquid we can go for the tank. In both way we can get idea regarding the peripheral view and the size and shape which we have evaluated by using the particle size characterization by maybe the laser diffraction technique which is widely used. That is on a larger scale. For individual particle, we have to go for the SEM or TEM. In many cases, if we are creating crystalline nanoparticles like NLC, which is a subclass which has been created, nanoliquid crystals or crystals, then we have to go for the crystallography. Or in special cases, if any transition are there into the lipid on a solidification, that can be confirmed with using this technique. Either maybe go for the DSC or using the XRD. Any changes are there into the structural part or not. Now coming to the application part, it is widely explored for improving the bioavailability of the drug, where there is a poor viability are there, which is usually classified as below 60 or 50. That means if I am delivering 100 mg of the drug, only 40 or 50 mg is giving a pharmacological action. So I have to deliver more amount of the drug, where there is a chances of higher side effects are there, or the cost which is required for that drug will be going to rise. Especially if the product is high end, that means the anti-cancer molecule. Like somebody has told that even one injection is ranging from anything between 10,000 to 20,000. In that case, if you are reducing the total amount of the drug, we are almost reducing the cost also. And on either part, we are also improving the safety. So there is a both win-win situation are there for patient as well as for the industry as well as for a social cause. That by simple addition or technology part, where drug dose can be reduced, we can reduce the overall cost also. And usually the oral route is most acceptable, so that in future, that even though many of the injectables are there, are slowly started to develop into the oral formulation also. Even that phytopharmaceuticals are there, like curcumin. If you go through the market for cancer patients, even nanoparticulate curcumin tablets are available in market. That is already available in India. That has been given as a supplement therapy to the chemo as well as the radio cancer patients. So it is not new. The nanotechnology is well established, industry has accepted, even into the market it has been well accepted. Maybe the cost is one of the challenge which will be going to come up. Similarly, I cannot able to read some of the examples are there, like one of the more example which has been given for parenteral, that has been with the one more naturally derived agent, synthetic one, that is a resveratrol, that has been for improving the stability, maybe for, as sir has told, if the molecule is genetically derived, structurally too large and unstable. Like one of my scholars, she is in INTAS, so she is working with the clotting factor. And the stabilities are in days. Now to make them long circulating and effective, even into the injectable formulations, they have been struggling. They have been making the different kind of the complex conjugation which has been reversible in nature. That can be encapsulated into the such a solid lipid nanoparticle and can be delivered based on the specificity where this lipid can bind to the targeted site and release the drug at a desired rate. Similarly, for many of the cancer and the molecule which has been used into the Alzheimer's or to the migraine, where there is a permeability issue, BBB, we cannot able to cross it easily because if the water solubility is too high, the drug cannot permeate through the BBB. So for that purpose, the drug are encapsulated into the solid lipid nanoparticles and delivered for a circulation. As it goes into the circulation, they may cross the BBB and deliver into the brain. We can go for the nose to brain. We can convert these solid nanoparticles into the nasal delivery system and can be enhance the overall accumulation into the targeted site. So cancer, it's a although not a new disease, but if you look into the numbers, I don't know about this reason, but in Gujarat, if you go day by day, there are a number of the cancer hospitals are increasing. If cancer hospitals are increasing, that means the requirement, the patient are increasing. Maybe there is lots of talk are there that awareness of diagnosis is increased, that's why the patients are coming up. But on either side, there are not many treatments are there which has been available. So that if the primary or secondary stage of the identification are there, diagnosis are there, they are curable. Maybe not 100%, but you can improve the life cycle of that patient to much more extent by using a specific targeted drug delivery system. And if you see, I personally observed with one of my family members, we have an older mindset that as soon as the chemotherapy starts, mostly the patient have issues with the lot many side effects like hair loss, diarrhea, vomiting, but as the new targeted injectables are available, they have to be with the less side effects. The molecules are same, only the technology is changing. A technology with the specific excipients which drive them to release the drug at the targeted site. If I have a brain tumor, drug must be released at my tumor site only. That is specific targeted site. It should not release anywhere into the liver, kidney, heart, lung. If it is exposed to that organ, it will start to create a multiple organ failure or side effects. Because these all drugs are nothing but the reducing the cellular growth. The metastases are there, which they usually kill the cell. So for them, where there is no solution are there, like sir has told that we are not synthesizing the pure NCE. Okay, we took a time, but if even though somebody has synthesized the NCE, but it has been having the side effects, like remdesivir. All of you have heard remdesivir. Corona time we have used, but still the patients are suffering from its side effect. The molecules was rejected because of the side effects. But emergency approval are there, there is no other solution are there. Risk-reward ratio is higher. In that case, we have to go for the reward, and we have approved. Now nobody is using it. But if somebody says that I produce a formulation which has been having a zero side effect of this drug, that will become as a blockbuster. So only maybe some student is asking for MBBS. We say we have a good facility infrastructure for producing a novel dosage forms, characterize them, market them. We can go for even clinical trials, BA/BE or even the clinical trial later on. And that's a huge market. Although we could not able to afford the cost of NC synthesis, where there is a chances of failure are more. But if you took the success ratio of MBBS is much more. So that's why it is not like that the investment is not there. They are investing, but the profit margin, risk reward ratio towards industrial concept is higher into the MBBS. And that's why many companies are coming up with the F&D center and R&D centers where they are slowly evolving of the new and new technology part. Even maybe sir has told about 3D printing. Maybe the future will be there. That is, you can adjust a dose. Likewise, you can produce such lipid nanoparticles also. Maybe dots are there, rods are there, nano dots, nano rods, nanofibers. So these things are synthesized that can be precisely synthesized by using a technology like even 3D printing or maybe some other techniques will come across in future. So brain targeted. Now we know about the few diseases only. Even in India, Alzheimer is not disease, Parkinson is not disease. Even nobody goes for the identification of any kind of the genetic defects into the neuro. They consider it as okay, hota hai, it may be there. But if you go towards the Western countries, as sir has told, this has been becoming as one of the challenge as the cancer are there. We have been more focusing on these till date towards some of the communicable disease, malaria, tuberculosis and other. Then we have slowly started to evolve onto the diabetes and then the hypertension. But there are a lot many neurodegenerative diseases are there, which is usually start to appear at the lateral age, 40, 50, 60, and then they shows their symptom. So where again, this kind of the technology, where high dose is required to be delivered in oral route for effectiveness, we can reduce much more, even 1/5, 1/10 of the dose may be effective by delivering into the solid lipid nanoparticles. Even a few of the preparations of Sun Pharma is available, which has been by nasal route or ocular route. Even cyclosporine is available. So there has been nano emulsions are there, and slowly you can convert it to the more and more number of the compounds for lipid-based formulations. Along with the delivering or crossing the BBB, these selective lipids, which can be handpicked, customized as per the receptor targeted site. Like one of the polymer that Professor Wong has told that chitosan and the alginate, cationic and anionic polymers are there. They have a charge on them. So if you deliver them on positive side towards the blood-brain barrier, they may have a particular sites where they can bind and release the drug. Even similarly, maybe meningitis, there are many works are going on that. Nasal drug delivery system, a liquid are difficult to maintain into the nasal cavity. So what you can do is, you can deliver into the mucoadhesive, maybe polymeric or maybe the adhesive form. And these solid lipid nanoparticles can be easily transported through the olfactory region and can deliver the drug to the brain. So like oral, you can go with the nasal cavity or brain targeted. It is not used much for the simplest disease, but for many of the diseases in future you can try because it's non-invasive in nature. You don't have to inject. But although the effectiveness is equal to the almost parenteral route. Like you have seen of the many of the narcotics and opioids which has been taken by the nasal route. They'll give quick onset of action. This is one of the class where if you can convert them into the depositive solid particles, there are sprinklers are available which you can use for spraying a potent medicament for effective therapy. It has been explored for the delivery of the vaccine also. So in future, you may see that the more number of the biologicals which can be delivered by this route. A pulmonary, lots of talk are there, high perfusive organ, there are blood perfusion is very high. So if you are able to target that side, the chances of getting the drug into circulation is very quick. But there are their own challenges are there. How to reach, maybe Professor Wong has told that 5 micron or below that. Maybe size is one of the limitation are there. You have to go up to 5 micron or below that. If you can convert it to the lipid nanoparticle which is solid in nature, you can simply use the DPI. Nowadays the dry powder inhalers are more popular from last maybe 10 years. There are Rota inhalers and other brands are available. So that it can be inhaled, the drug will be goes to the exact targeted site. Now, if I have any infection at the lung site, maybe bacterial, viral infection or maybe like cancer, I have to treat that by delivering the drug to the targeted site. Although how much amount I am delivering by the IV or oral route, that may not be enough for reaching to the lung site compared to if I directly deliver the drug to the lung site. So pulmonary, which is a breakthrough, we have a technology for delivering that, either in a liquid form as well as for the solid. Whether they are in nano or micro, we can deliver. Only thing is we have to convert the drug which is available to the such class. And the technology is not that much costlier also. Only as somebody is told that patent expire. Okay, things are there. What we have to do is we have to prove the superiority. No, after expiry you can even have a similar effect. You can go for generic formulation. There is no requirement of going to the larger scale. Even if it has been many of the class are there, especially it has been nowadays given for the waiver also. Ocular, one of the fascinating site where you require to reduce the hypertension, that is ocular pressure, to the many other infectious diseases. And even many of the genetic diseases are there, which is known to be uncurable, but that can be controlled. So the further degeneration can be stopped. So the improvement, like sir has told that one of the drug which has been repurposed, bezetonic, so that can be used as a nanoparticle and can be delivered, which can be absorbed through, because the permeation is easier. It can cross the cul-de-sac and release the drug at the site, and slowly, maybe for 1 day, it will remain 2 days, 3 days. So the long-lasting effect are there, so that you don't have to put the many drops into your eye. You don't have to use it repeatedly. So the control release, sustained release formulations that can improve the overall therapeutic benefit to the ocular disease, especially in case of the posterior segment. So these are some of the available research which has been done on to the solid lipid nanoparticles with the method and their characterization and evaluation which has been done. These are some of the kinetic data are there, because we know that the in vitro is very easy, but these are some of the summarization of the kinetic data are there which has been administered and how effective they have been found. So if you see on to the right side, they have been summarizing the how much fold of increase into the either residence time or the bioavailability are there, or either the effectiveness are there, or maybe the cellular loading are there. So it has been ranging from anything between 2 to 4 or 5 which has been found at the laboratory scale and which can be expanded into the furthermore. So these are some of the summaries are there which has been collected from the last 5 years of the research, and we have published one review on that. These are some of the patents are there. There are not many patents are there. If you go through the even Indian patent, there are day by day the number of these formulation based patents are increasing. Now coming to the research segment, for solid lipid nanoparticles, we have done few research on the anti-cancer, then antifungal molecules, as well as onto the various kind of the drugs which has been improving the bioavailability into the body, like exemestane hydrochloride. We have explored with the liposome, solid lipid nanoparticle and the nanoemulsion, and we found that the solid lipid nanoparticles have highest stability and have equal absorption, permeation compared to the nanoemulsion. Similarly, for ocular delivery, we have used a caspofungin, which has been used for this ocular fungal infection which has been caused during this corona. So that drug, which is highly potent, we have improved the therapeutic benefit to the 2 to 3 fold by preparing into this solid lipid nanoparticles, and stability is improved to a much higher level. It has been more than even 6 months. The drug is stable at the -10 to -20 degree. So normal storage of that drug is into the sub-zero level, which can be stored at 4 degree to 8 degree by preparing into the solid lipid nanoparticles. A few current projects are going on to the, one is on tadalafil, which we are working on to the delivery for pulmonary hypertension, which is preparing the solid lipid nanoparticle using Compridol 188. So this has been the current research which has been going on this field. So as far as the larger scale production are there, still the challenges are there. We have to work on to the stability, maybe to improve to the multifold, that means to like solid oral, 3 years, 5 years of expiry. Scalability, large scale production is still difficult, but on either side there are many discussions are there. If requirement will be improved, the scalability will be automatically come up. There are instrumentation technologies which can work on, but only thing is the demand. How much demand is there? Usually the demand of such high-end molecules are 3,000, 5,000 vials are there per year, maybe 10,000 vials, where the scalability cannot be expanded to the larger scale. But as there is a day grow, if the demand will be higher, that will be going to scale. Regulatory hurdles, sir was asking about some conjugation and other thing. So if you are mixing too many things together, whether they are safe, effective or not, that we have to prove. So by using various kind of the lipids which we are maybe synthesizing from the natural or synthetic origin, combining with them, purifying them, using them, whether they are safe or not. So we have to follow the whatever the regulatory hurdles are there, and what that makes as one of the limiting steps is you don't have versatility. Like you go for the tablet, you have a lot many binders, lot many excipients are there. But over here, you have to play with the body. Many of limitations in selecting the lipids. You have very limited choice are there, where you can either go for the Gatfose, Evitec and Merck. These are the 3 largest manufacturers are there of the lipid. So they have owners and they have been reserving their technology. So the cost is also slightly higher on either side. So overall outlook, future, you can use it for personalized medication, small dose to the large dose, you can use it. Genetic or biotech derived products, you can encapsulate, you can deliver by using successfully encapsulating into the solid lipid nanoparticles. In future, it has been explored for the diagnostic purpose also by using bioimaging. So you can have some regulatable compounds are there, which can be safe enough, which doesn't produce any kind of side effect to the patient, which can be have more accurate binding sites. So as much as higher the binding sites are there, your identification will be more accurate, precise. And the sustainability, which has been the talk of town if you are going for the organic, it can be produced as non-organic flavor. This has been the Scopus AI produced word art, which has been giving the idea regarding the what research and keywords which has been frequently coming across. Once I write solid lipid nanoparticle. So even if you see there are few things are there which has been non-pharmaceutical. Functional foods. Somebody maybe they're from the Nutra and the food sciences. Functional foods are there. We know that the nutritions are there which we are taking every day. We require in micro level. If we have synthetic or maybe the synthesized compounds are there which has been similar to them, we can use it to deliver in effective manner. Simplest and widely used such things are the volatile oils, which has been for the either to the inhalation therapy or either to the topical therapy. If you can improve just the stability, volatile oils they are generally evaporating as you open. So you can just encode them. You are providing some simple coating and they are stabilizing. Then effectiveness, therapeutic benefit may remain same, but on either side they have been more stable, long lasting. Then denaturation, whatever the degradation kinetics are there, oxidation and other things can be reduced. So in future we may have a specialized functional foods are there which can be used for the nutrition purpose or either the supplement therapy along with the normal therapeutic benefits. Even it has been widely accepted to the first keyword if you see on to the left side, cosmeceuticals. It's you can say the money making market is towards the decoration only. So the industries are producing day by day different kind of the cosmetics with the incorporation of the different polymers, different oils, and on that side again these nano formulations can increase the therapeutic benefit. Either they can give the micronutrients to the skin and increase the cellular performance. They maybe reduce the simple disease like acne, where there is a difficulty, still we are facing some difficulties for delivering the drug to the site and controlling them. So future will be there, maybe even we can reduce the production of the melanin also in future, maybe by such mechanism or maybe some kind of the nutritions. So as we grow, the demand for technology are there, and this is the technology driven formulation only. The drug will be same, only you are changing the concept of delivering them to the required site by using the proper combination. Although the cost is there, but as the demand is growing, we have to change our conceptualization and the need are there, so we will going to evolve off. Maybe 10 years back only few products are there of the liposome, nanoparticle, solid lipid nanoparticle, nanoemulsion. But within last part of the 10 to 20 years, more than around 200 plus products are launched, which has been widely used. And although they may be under the scrutiny or maybe under phase 4 and observed, but there are many molecules are there which has been gone to dustbin, which has been started to repurpose by using this technology. So we can use the available resources. Although the molecular synthesis will take 20 years and nobody could able to make the safe and effective dosage form. You can take it from there. It's a ready-made available. You try different technology, use the NDDS concepts, work on that, deliver in safe and effective way for betterment of society. So, thank you, Dhanyavad, for patient listening. And again, I have to catch a flight. Maybe you have to a bus or something like that. And I got a coffee over there before coming over here. You got or not, I don't know. So, being a fresh till the evening from morning 9:30 or 9, it's a great power. And maybe I'll thank the Adamas University to organize such an event for the students to learn, to demonstrate their research, to be competitive, and hopefully after 10-15 years I may see somebody of you having your own research center, somebody has maybe general manager of F&D, maybe somebody is having business development or marketing segment of the novel formulation excipients. So you are the future. If we say in the term of our Prime Minister, the knowledge is there, but it requires to disseminate. So you are the main carrier of that. And now technology is so fast. Even somebody speaking over here, people start to use the ChatGPT and start to using Google. So knowledge is not very difficult to gather. But whether you are using them day by day, in routine practice, at laboratory level, for your purpose, for your surrounding people, for the society, that's one of the main cause of research. Many of the funding, as sir has told, maybe the Western countries are funding. They have been funding to the disease which has been more prevalent towards their people. So our focus should be towards our problems. Like till, how many of you understood that tuberculosis is gone from India? TB चला गया कितने मानते हैं? नहीं गए. कितने साल हो गए? What? We had been trying for simple TB and malaria to be absolute out. All the contagious disease, communicative disease. So day by day, the need are not only for the industrial aspects. There are a lot many things are required for the social cause. What the end users require. What are the ground level people's need are there. The country invest into that. If you want to fund, maybe some of the faculties are there, or maybe some students are PG, PhD are there. If you want fund, higher amount of the fund, apply for the proposals in that direction. Like sir has told, they got 100 crores. Murthy sir has told in morning. 100 crores for what? Phytopharmaceutical characterization. So what we have large natural resources. The limitations are there. Okay, no, we cannot go for the more of the biologicals and NC. Okay, but what we can do, we can successfully go on to the NDDS path. I have more than around 25 students who have been working into the F&D at various levels till the VP. And they have been grown like nothing. They have been paid highly, even more than 10 or 20 times than me. Staying in Gujarat only, maybe staying in Maharashtra, not far away. There is a huge demand in pharma, nutrition, cosmetic, or science-based product in market. Only thing is whether it is competitive. As a survey bola tha, whether it is equal or more than that. If you can able to prove that it is slightly above than that, it will be there. If I can able to synthesize a molecule which is better than paracetamol in terms of low dose and better therapeutic index, safety, that therapeutic index is much wide. obviously paracetamol will be going to absolute out in 5, 10, 15 years. When the diclofenac and aceclofenac synthesize, many have thought that ibuprofen will be having a no future, but still it is there. They have their own advantage, they are carrying with them, although having a high dose, they are there. Maybe used in a lesser amount. When nimesulide came, I was in Alembic at that time. When nimesulide was introduced and celecoxib and rofecoxib were there, we used to market them. Many had told that there are no antipyretic drugs now required because nimesulide is enough for both pyretic and the analgesic. But after a few years, it's gone. Safety, efficacy. Although the molecules are released, approved, they have been under watch, reported, they took back. So if you could able to use the technology to such molecule, आज अगर कोई बोलेगा कि if I can make a same nimesulide formulations for pediatric, you will again start to use. It's not like that. But you have to overcome the whatever the problems which has been associated with that molecule, which is reported. And if you could able to solve these problems, you can use that technology, you can sell that technology, or you can earn a royalty also, which is also a part of the patent. You can sell or either earn a royalty lifetime, or your maybe children and grandchildren will get a royalty. So, thank you again, and
Thank you, sir.
I'll thank the organizing team along with my good friend Professor Biswajit Basu for inviting me. Although maybe we have some hurdles are there for maybe I could able to attend or not, but I made it and you have made it. My email address is gone. So any students have any query, or you can able to ask over here or not, you can drop a mail to me. I'll be available on call or either on mail also, or you can get it from the organizers.
Thank you, sir. Definitely the students will have a lot of questions. The success of the session is that we have started your session with the number of participants and we are ending your session the double the number of participants in the audience. That is the success of today's session. That's it. Before inviting a few queries from the audiences, is customary to summarize. I have started that sir is actually the master who works in the drug delivery systems based on nanotechnology, and sir has proven it lucidly in a lucid language regarding the importance of solid lipid nanoparticles, the way it was prepared, the objectives to prepare solid lipid nanoparticles, the advantages and the benefits, explained in a lucid language the preparations, that is making formulations using solid lipid nanoparticles, characterizations of the same, and the delivery systems that can be utilized using SLNs to target brain to prepare vaccines in modern days. Sir has also pointed out the areas where a nanoparticle can be utilized, like pharmaceuticals, not only limited to pharmaceuticals, but nutraceuticals and cosmeceuticals also. I am sure that right at this moment, majority of the M1 projects are being conducted on the preparation and characterizations of SLMs. And most of the PG scholars also in the area of pharmaceutics here in eastern part of the India also are working on this preparation and standardizations of SLMs. It can be an important delivery system when we are targeting a specific site and objective-based targeting of the molecules. As I said that there is no dark knowledge in the generation of AI and GPT, but it reminds me a lot that there is still the utility of the faculty members on the dais and from the dais to interact with the eyes of the students, to interact with the eyes of the audiences. The importance will definitely be there. Artificial intelligence has been prepared by human intelligence, and I am sure that human intelligence will win over artificial intelligence. I have a small query, sir. It's a very simple one. What is actually basic differences between liposome and then nanoemulsion and solid lipid nanoparticles. I think there are marginal differences between liposomal drug delivery and solid lipid nanoparticles.
Liposome may be slightly larger, but that not much difference are there. But it is lipid which is in a unstable stage, which is liquid. So lipid, it's like bubble. So it may break out. They are unstable in nature. To stabilize them, we have to go for the addition of the certain agents, and then we have to lyophilize. So it requires additional stage process as far as the cost is concerned. In market, there are not many liposomal formulations are there. If you compare the cost of SLN to the liposome, the SLN will be always lower cost compared to them. Then the choice of lipids. For making a liposome, there are very limited number of the safe and effective lipids are there. Whereas on either side, we don't require lot many selective or wide variety of the lipids are there. The processing part, again, there is a high pressure is used in both, but at the end we have to stabilize and allow them to aggregate. So the size should not be grow to a larger level. On either side, nanoemulsions are well established and acceptable. Again, it is a liquid product. You have to carry, you have to deliver in a liquid form only, which may be acceptable, which may be difficult to deliver to the many patients. There is a taste issues are there with the nanoemulsions. So if you are using oil component, they may be giving a different kind of the taste and smell. They always supply in a liquid form, so the acceptance ratio compared to solid are much lesser. Now maybe sir has told that we can convert it to the solid also. So the emulsions can be absorbed onto the some absorbing agents like the silicon dioxide and the derivatives of them are there, which we can absorb and we can convert it to the solid tablet. But again, it requires